Good afternoon. I'm very excited to be here, and thank you for staying for the last talk. I really appreciate it. Sorry that the title of my talk was not very informative until really recently. I was actually hesitating between the presentation of two different work, both on image segmentation, and I attempt to say that they are unrelated, but it turns out that they are related, but in somewhat a confusing way, so I hope I can go on with that. In the first one, I revisit the level set approach for multi-region segmentation, but I will present a different view of the good old level set function. The second work, I will use a logistic regression function. I will not use level set, and in a sense, this is machine learning work for image segmentation. So let us formulate a problem. So we have an image defined on a domain. It can be 2D, 3D, 4D, whatever. We want to partition the image space into disjoint region, and we could define for this or find for this labeling function. We can use the one-hot vector in order to classify or assign each of the image voxels or pixels into a label where we can define the all-zero vector for the background. So we wish to find regions denoted by omega 1 till omega m, and for the background without loss of generality. And we wish them to be mutually exclusive and to cover the entire image domain. That is, we wish not to have vacuums or overlap. Now, when we do deal with binary segmentation, we can solve it quite elegantly. However, when we have more than two regions, we may have some difficulties, in particular in those junctions, those voxels or pixels that on the boundaries of three or more regions. It seems to be sparse, just sparse set of voxels that has this problem, but when we deal with 3D imaging, and I do a lot of brain imaging, and we have 3D image domain or sometimes 4D image domain, we can have regions or interfaces that define the boundaries between three or more regions, and this is a little bit more complicated. Now, multi-region image segmentation has been dealt before, and there is a long list of work, and just to mention very few of them, there is the work of Zhu and Yule, region competition, which turns to be really influential in the field, and then Vizi and Chan proposed multi-region level set segmentation, where they use log m level set function in order to partition the image space into regions. Later on, there are other works, one of them of Brox and Weikert here. Jung and Chan proposed multi-phase segmentation, and recently, very recently, Ronnie Kimmel and his colleagues proposed to use a single level set in order to partition the image domain into M regions. Now, I'm not going to propose an economical way. I will use M level set function or M-1 level set function in order to partition the image domain, but I will present a different way to do so, so this can be solved elegantly. So I would like to start with a Bayesian presentation to the image segmentation approach. So we assume that the known variables are the image intensities, and we would like to maximize the joint probability of the image intensities and the segmentation in order to find the unknown variable, the labeling functions, and the model parameters. Now, I do often deal with brain imaging, so I can assume that, for example, the model parameters or the intensity parameters can be modeled by a Gaussian mixture model. So in that case, for example, if we want to classify an image into several regions, I would assume that each is modeled by a normal distribution with parameters that are unknown and we wish to find throughout the segmentation process. So our problem can be boiled down into a maximum a posteriori, solving a maximum a posteriori problem to find the segmentation, and a maximum likelihood in order to find the model parameters. And we can do it by an alternating minimization approach or maximization using, for example, EM. Another way to deal with it is to construct an energy functional. Here, if we usually we assume that the image pixels or the voxel are independent, and we look at the product of the probabilities or the sum of their log. And if we wish to solve an energy functional, then we can replace the sum with an integral. Now, using the level set approach, what we wish to do is to replace this gamma, the segmentation, by a level set function and then solve it using, for example, gradient descent approach. Using the Bayesian formulation, we can actually partition the functional into two terms. The first one would be the image likelihood term or the data term, and the second one could be, for example, a regularization term. Now, for the level set function, I would suggest to use a probabilistic approach. So, for example, if you want to partition an image into two regions, the region of interest here marked in red, and the background, W out, what we usually do with the level set formulation, we define a sign distance function, and we evolve it using calculus of variation. But one nice way to do so is to apply a step function, a Heaviside function, or a sigmoid in order to do it a nice and elegant way. And for example, Chanel-Visi in 2001 suggested to use arctangle sigmoid to do that. What I propose here is to replace this sigmoid by a logistic regression function. Now it's similar, behaves similarly, but once we do that, I can assign the Heaviside of phi of a particular voxel x the meaning of probability. So note that the boundary pixels of voxels, the zero level set, have the probability of 0.5 to be part of the region of interest. So this is the probabilistic formulation or the log-out formulation. And if we have this formulation, then we can consider our level set function as the log of the ratio between the probability to be part of the region of interest over the probability to be part of the background weighted by epsilon, where epsilon here determines the smoothness or the slope of the sigmoid. So having this, so for example, if we play with that epsilon, the slope of the sigmoid, this can have the notion of probability or the uncertainty or certainty of the boundaries of a particular regions. What do we really do? If we take the functional, apply gradient descent formulation, in order to do that, we differentiate the Heaviside of phi, we get the delta function, and if we do this logistic regression sigmoid, we can have this nice property. The delta of phi is actually minus Heaviside of phi, smooth Heaviside of phi multiplied by Heaviside of minus phi. Now, this is for the binary case, well-known case. I would like to extend it into the multi-region formulation. And in order to do that, instead of using logistic regression sigmoid, I will use the multinomial sigmoid or the softmax function. So if we do that, then phi, our level set function, can be defined as the log of the ratio of the probability of being in part of the region of interest over the probability of being part of the background. So the heavy side, or the probability that a voxel X belongs to a particular region, is the exponent of phi I over 1 plus the sum over the exponent of all the other phis, or level set functions. So now the heavy side get a different meaning. Rather being specific to a particular level set function, we can tie here all the level set functions together. Now this is really important if you want to evolve the level set function and eventually make sure that they cover the entire space with no overlap. Now we can do the same for the heavy side that is represented by the background. And if you look, so 1 minus pi get this formulation, which I define here by p bar. So this is our heaviside function. If we want to differentiate in order to derive the gradient descent equation and get the delta, or the smooth delta, and here I use heaviside SM for the heaviside of the softmax, then we get this term, which turns to be, as in the binary case, heaviside of phi I multiplied by heaviside of minus phi I, or pi multiplied by pi bar. This is not a new result, but it's new in the context of level set formulation. Now the nicest thing, since every level set function, or every heaviside of a level set function, actually embeds together all the other level set function, if we want to differentiate phi I with respect to phi j, we can get this term, which is in a way can be interpreted as a repulsion term. So now we get the repulsion term really elegantly from the derivation. We don't need to just plant it in. Okay, obviously delta II is equal to delta I. Now for the background, we can do the same derivation. Now once we do that, we can redefine the segmentation functional. So let's have a look at the image likelihood term or the data term. So we want to generate a functional, and minimizing this functional with respect to each of the level set functions will ideally give us the optimal segmentation. So if you look at this functional, and you look at the function G is the Gaussian. So for example, for the binary case, if you assume that each region, the intensity of each region is modeled by Gaussian with variance 1, you simply boil down to the known data term from Chen-Vesey functional. Now this is for multi-regions, and we do not necessarily assume that each region is modeled by a single Gaussian. So if we do that, we can substitute the probability of being in the region of interest or not in the region of interest as following. And this is actually the heaviside, or the softmax heaviside of phi I, and the softmax heaviside of phi m, which define the background. Okay, the regularization terms, and here I heard a lot of other way to do regularization. We use here the standard formulation, although this could be in a straightforward manner and be adapted. Takes the following form, again using the softmax heaviside. Now for the gradient descent, we get this following formulation, and you can see here that the delta term is multiplied by a repulsive term, and this is derived really elegantly from the formulation, and this would allow us to generate a real competition between different regions in a way that eventually we get segmentation that would cover the entire space with no overlap. I will show some results. I work with brain imaging, and here you can see multimodal brain imaging. So we have 4 modalities. So in fact, we can say that our data is 40. So each modality has its own distribution model, but altogether we can look at it as a multivariant Gaussian distribution. And after convergence, what we get, if we look at the heavy side of phi of each of the region, and here I partition the white matter, gray matter, CSF, and the tumor region, you can see that you can get actually a probability map. And doing the maximum a posteriori over all this probability map, we can get the final segmentation. Now I have another result just to demonstrate the main idea. So here the aim was to partition the tumor into 2 regions, and another region would be the healthy brain tissues. And again, this is 3D, actually 4D, because we have a few modalities. And you can see this, the raw data, the source of the raw data is the Boris challenge data sets. So by this I end up my first talk, and I will move on to the second talk. In this talk, I will consider brain segmentation, and this is very specific to brain segmentation. The former talk and the former work could be applied to any image. So this is a healthy brain image, no tumor here, and it is really structured. So adult human brain look quite similar. And when we want to partition the brain images into, say, gray matter, white matter, CSF, brain tissues, we would like to use the structure as a prior. This can actually leverage the performance of our segmentation, because without that, one might have a lot of problems. Why do we want to do that? Shape analysis, aging, etc. So we can formulate the problem in a similar manner. I is defined on the image domain, which could be, for example, 3D. We would like to partition the image into L disjoint regions of interest. And again, we can solve it using the Bayesian approach, where we have the image intensities. These are the observed variable, and we would like to find the segmentation. It's slightly changed the notation, so I used gamma there and y here. And here is the, we can again model the intensity distribution by a mixture of Gaussian. So again, if we can go on and look at the joint probability of the image and the segmentation in order to find the model parameters and the estimated segmentation. Again, this could be partitioned into a likelihood term and a prior term. So in the previous work, I took the prior term as our regularizer, and here I would like to embed information I have from other brain images. Now, the easiest thing to do, if we assume that the intensity of the images are distributed, can be modeled by a Gaussian mixture, is to look at the mixing proportion and define the prior probability accordingly. However, this may not be sufficient, and we would actually like to replace this by some spatial term or spatial prior, such that for a particular voxel, we would know the distribution of that particular voxel and not in general in the image. In other words, what we wish to know is some function, kappa x, which will be the mixing proportion of that particular voxel in the image. So obviously, the probability of being gray matter, for example, is higher in particular brain region than in other brain regions. How do we get that? A common way to do so is to construct an atlas. Okay, so people do use, take a training set of images, brain images, 3D brain images, and hopefully having a clinical expert, they can have a segmentation of the image into the region of interest. Once the segmentations are known, they can be taken, be averaged, or some other form of statistic can be extracted, and here we can have the mixing proportion for each voxel in the image. Obviously, these images has to be aligned. Now, this approach, the atlas-based approach, very popular, very efficient, very economical. So, for example, in terms of memory consumption, if with the number of voxels in the image, the memory would be just the atlas, a single image. So we can summarize all this large data set, which could count hundreds of images, with a single image, a probability map. Okay? In terms of registration, all we need to do is one registration, simply align the atlas into the image to segment. However, this is not so informative because we take into consideration the labels of the image produced by the expert, but we do not take into consideration the image intensities. And the question is, can we do better than that? Okay, so we have very economical way to store large data of images to generate a prior, but the question is this prior informative? Now, in recent years, a new approach has popped up, which is called a multi-atlas approach. It has different names, and the idea is the following. Again, we have a large training set and images and associated labels, but we keep it as it is. Okay, we store all the data, and once we have a new image, a test image, we use just part of the data or a weighted summary of the data in order to generate the atlas. Okay, so what we do, we tailor the atlas to the image. Okay, so once we have a test image, a novel image, we do registration of each of the images in the data set into that image in order to pick those images that fit the best the test image. Or to do some weighted sum, other statistical form. This obviously requires a lot of work during test time. We need to do a lot of registration, and at the same time, we need to store V multiplied by L multiplied by N memory. So there is a huge memory consumption. Okay. So this may not be good enough, and the question is, can we do some compromise? So the classical atlas approach, it's fast, the test time, low memory consumption, not very informative. The multi-atlas approach, fast test time, no. Low memory consumption, no. But better, more informative. So can we do a compromise? In order to do that, what we propose is a new form of atlas that we term the atlas of classifying. So the main aim is to store less data, do some less work during test time, and at the same time be more informative. So can we store more informative information having a large data set? So this is the main principle. Okay, let's have a look at a particular voxel along our training set, and let's construct a vector of the image intensities. Now, having the labels of this vector, we can associate an intensity or scalar to each of the label and construct a sigmoid that will store the information of the entire vector. So in fact, what we do is we try to learn the shape of the sigmoid, or in other words, learn the W, the weight, and the bias term that best classify this vector of intensities. Okay? So we can apply this to all the pixels, voxels in the image, and generate an atlas of classifiers. So rather than storing just the probability of being part of each of the classes or each of the tissues, we store a sigmoid or the parameter of a sigmoid. In fact, only the bias and the weight W for each voxel is preserved. So rather than storing one parameter probability, we have now two parameters that take into account both the image intensities and their labels. So we now do not need to store all the data, just these two parameters for each entry. In fact, image intensities by themselves are not strong enough. So if you take two T1 weighted images, for example, even if taken by the same scanner, the intensity range of gray matter tissue, for example, can vary. This depends on many parameters, including those related to the patient, say patient weight. So rather than relying on the image intensities themselves, we would like to do something or to extract some features. Now, what people usually do, they normalize image intensities, but we wanted to avoid that. So in order to overcome it, we define, instead of using one scalar, the image intensity, we look at the intensity distribution of the entire image, and we replace each scalar, each image intensity, by the probability of being part of each of the Gaussians. So now, rather than having one scalar to present each voxel in each image, we have one vector, and our sigmoid or logistic regression function change accordingly. Okay, so we replace the scalar by a feature vector. So we have m features for each voxel and entirely m multiplied by n features, and the number of weight change accordingly. Still, the number of feature m is much lower than the number of images in the data set. So we are more economical, but yet more informative. So rather than having a scalar, which is W, we have a vector where W0 is the bias. Now, the sigmoid or the logistic regression function is good for the binary case. We want to do the segmentation for multiple regions, multiple tissues. So we use again the multinomial regression function or the softmax function. So in the previous work, I've shown how to replace the heavy side of the level set function by a softmax, but here we use it to represent the prior. So we have a different representation here. Okay, so once we have the logistic regression function, we now have a larger set of weights. So for each class, we have a vector of weights. So suppose that we have m features, L labels, so we have m multiplied by L features. Okay, so our framework is composed of training and testing. So the training, what we do is simply we solve this objective function, we do the optimization. We usually do this regularization. We were working on this but ends up using this one, a simple one. We could have used something more sophisticated, I know. And having this objective function, we can derive the gradient descent equation. This is, I guess, very familiar for those of you who work with a neural network, for example. So we do the gradient descent in order to learn the weights of the sigmoid or the logistic regression function. And at the end of the process, once we have the weights, we can use them in test time. So this is a convex optimization function. It converts really nicely, and we eventually are more informative. How do we initialize the weights? Simply by white noise. So we start like this at the beginning in the first iteration. After one iteration, is how the weights look like. Okay, so you can see here the weights for the bias, the white matter, the gray matter, and the CSF. And after one iteration, it has the form of the brain. And we can go on after 5, after 10. It converts really fast. Okay? So these weights actually embed our entire data sets. Now, one question people may ask is, why not using a tissue classifier? So we could, example, define a classifier. Many people do that for each of the tissues. So here is we try to answer this. So here you can see the white matter feature weight for the white matter tissue weight, and this is the label. We can superimpose, and we can look at two different voxels, the same, more or less the same region of the brain, but you can see that there is a significant difference in terms of intensity. We can actually quantify that. We measure actually the mean and the standard deviation of the weights distribution for each tissue, and you can see that there is a significant standard deviation, and also we measure it in terms of the entropy of the weight distribution. Okay, so there is a justification to do an atlas of classifier, that is to define a classifier for each voxel rather than define a classifier for the entire tissue. One more thing I should note is related to image registration. Obviously, we need to register the image. We do most of the registration work during training. So we pick one image and we do the registration with respect to that image. Actually, we do not use the image intensities, but again, the image features. Okay, so we extract the image features and we do registration, a pairwise registration for the entire training set based on three features, the probability of being part of the gray matter, white matter, and CSF, and we get different registration parameters. So in fact, we can use this in order to augment our data. So rather than having n training example, we might have n multiplied by n minus 1 training example, which is really significant. So it turns out that with this kind of augmentation, we could use very, very small data sets. Even 4 or 5 images may be sufficient. So just to go over, we have some work during training. We get the images and the label. We extract the feature. We construct the atlas of classifier using the gradient descent approach. But then at test time, it is really simple. All we need to do is to register the atlas of classifier to the image to segment. Once we did that, we can project it on the classifier and get the segmentation. So in order to test our approach, we used several publicly available data sets and use the dice measure in order to compare them. Now we need to show that we are better than the benchmark. And here you can see the dice score for the IBSR 20, IBSR 18 data set, and we compare it with few state-of-the-art method, and we show that we are better. So if you have a closer look at some segmentation results, you can see the original, the manual annotation that we compare to, done by expert, and our Atlas of Classifier. You can see that you can get much better, much more accurate segmentation than other approaches. Okay, and this is another example. Here I zoom in on the IBSR 20. What is nice about this framework is we can actually use one data set and test a completely other data set. So one example is that we apply the IBSR 20 on the IBSR 18 data set, but we can even do more than that. So one of the advantages is that we can do a cross-modality. How do we do that? So suppose that you have a data set of T1 weighted images of many patients. And now you have a test image of T2. With the Atlas of Classifier, you can use the atlas, the available atlas, in order to do the segmentation, although the intensity distribution are quite different. And you can see here some particular example. So what is the main idea? Suppose that we have a training set. This training set consists of T1 and T2 FLIR images. Suppose that our test is T1IR. We can use the training in order to do the atlas, and then apply the test on the classifier. And here are some cross-modality examples. Okay, and here you can see the segmentation. Okay. Obviously, there are some modalities that are more informative from the other, but still, this is a difficult task. So just to summarize, the Atlas of Classifier aimed to be more informative, yet much more economical and compact than the multi-atlas. Actually, when you think about it, it could be considered as a single layer neural network or a grid of artificial neurons. So if you look at this chart, so here is where I would put the atlas-based method. Here is where I put the very fashionable neural network methods, and the Atlas of Classifier is somewhere in between. Now, one question to ask is, why not using a neural network? So I can find a few reasons to that. Well, it might be that the result would be better, but our training is really fast. We do not need too many training examples. The nice thing is that we can do all these cross-modality things without too much training. And this is obtained simply by knowing our data, not letting the network generate a feature, doing it ourselves. Although we can, we can still use neural network and use the Atlas of Classifier as some sort of insight. That's it. Thanks.
Thank you very much. Questions?
What kind of registration are you using? It's like a non-rigid registration?
Yes.
This can be a hard problem, isn't it?
Yeah, this is indeed a time-consuming and a difficult problem. This is why the multi-atlas approach is really time-consuming, because you need to register for each test image. You need to register all the data, training data images into that test images. While here, you do only one registration during test time.
How reasonable? What's the variance of brain among the population? Are all the brains that similar that you can register them in a single atlas?
Well, healthy adult brain tends to be similar enough. Usually you do affine registration and then you do fine and diffeomorphic registration in order to improve that.
So is this for 3D data, I suppose, right?
Sorry?
This is for 3D data?
Yes.
Okay. So if so, how many examples, like in your atlas, it's kind of ground truth, right? So somehow, so how many ground truth examples are there in the segmentation of the brain that you can count on? I'm just curious.
Okay, so the IBSR is 18, has 18 images. So we use 17 to train, and then in a Lieberman out manner, we do the other one. 20 images. The cross modality, we have only very few images. We had like less than 10, and we still did it. So we do augmentation, we flip the brain, and we also use the trick with the registration, and it works. So we can do pretty good with a very low number of training samples.
So could you use that as somehow an answer why it can be done maybe with neural network, because like there's too few data, or there are people who successfully use this type of databases to learn neural networks?
What people do is actually they do not work on the entire brain. They do convolutional neural network, they do patch-based, they learn patches, they do a lot of augmentation, and still you need, I think you need, for these data sets, the reason I didn't show any comparison to neural network, I haven't seen a particular example.
And what's your explanation for why they...
Because you need a huge variety of data.
This, right?
For the cross modality, for example, you would need a lot of layers, much more parameters. So rather than so here, what we did is we used the fact that we know that brain intensity of brain tissue can be modeled by Gaussian mixture. If you use neural network, you simply fit in the brain itself with no prior and let the network run.
But would you say there is no hope for the neural networks, or like you'd say they just have to try harder, or like?
No.
I mean, this is a guess, of course.
You have to be patient. You have to have really efficient computers work on, or the alternative is to try maybe to make your neural network shallower, not that deep. Maybe there is no need to make them that deep.
Do you see a connection methodology-wise between your first part of the talk and the second part of the talk? So in the first part, you described the Gaussian mixture models and the EM algorithm, basically, with parameterization. And then in the second part, you also added this compression of your data. So that's kind of, I think, a compression of the covariance matrix that is usually block sparse, I think. And in the second part of the talk, you add basically the learning component. But the learning component is usually also in an EM algorithm inside by learning the covariance matrix or the first or second order moments. So I think it would be very interesting for the community to see the connection between EM-type algorithms, let's say statistical inverse problems, but on the other, in connection to deep learning methods. So do you see from your two talks how that could fit together?
Okay, so this is a really interesting question. I wish I had some more insight to say. I'm still thinking about it, but it might be.
I especially asked because in the second talk you compare in the results with the EM algorithm, with the Gaussian mixture,
Yes.
which was exactly what you used for the level sets, right?
Yes.
So in some way you compare to your own method.
Okay. Yeah, in some sense, I did this comparison. Not exactly, but yes, in some sense. Yeah.
Just a quick question. So in the first part, so for your Gaussian mixtures, how many modes do you need?
Well, it depends. I wish I could say that I can model each tissue by a single Gaussian. It's not always the case. For some data sets, you need a mixture of Gaussian for each tissue. And in some cases, there is a significant overlap. Well, it depends. This is something that you have to somehow learn from
You
your
would say
data.
mostly one, but sometimes 2 or 3. Is that right?
Yeah, it depends. It depends.
But never huge, never large.
Well, no, never huge. Never huge. It depends. If you want to model a brain with some kind of pathology, then you would need much more, because actually clinicians used to partition the brain tumor into edema and core and other regions, so you may need some quite few.
The reason I asked is sort of curious how it scales, like if you have more and more, sort of how the performance of the algorithm would be.
Okay, so obviously you would have more features. For the first part of the talk, this wouldn't matter because you would have multivariate Gaussian rather than in the second part, so you would have much more parameters.
Okay, so I'd like to thank the speaker.
Thank you.
Thank you very much.